The research Ben has carried out in his lab is nothing short of groundbreaking, and for his work he's really taking advantage of the full power of genome sequencing and the rapidly expanding data sets containing sequence from many individuals, from well-characterized human cancers and from other sources. Ben uses a combination of experiment and bioinformatics to identify genetic networks to understand why mutations are not randomly distributed and why mutations associated, for instance, with cancer are not randomly, are clustered at certain sites in an unexpected and rather surprising manner. Now, I don't want to steal his thunder. He's going to tell this by himself. But his work is really, as we think, of fundamental nature and has profound impact on our understanding of phenotypic variations, including human disease. And I would like you now to welcome our award winner, Ben Lehner. Please come up. Congratulations.
So, here you see, that's official now. Congratulations.
Thank you.
The trophy for the winner. Congratulations also from my side.
Thank you.
So, Ben Lehner.
Okay, and most importantly, thank you very much to the people from the committee for choosing our research and for Eppendorf supporting this thing. I think it is it's embarrassing to get these prizes, but it's very good for science to have this visibility, especially when you're young and doing basic research to be acknowledged. Okay, and now I'll tell you a bit about what we've done, and it's a kind of straight science talk, so I haven't really dumbed it down at all, so it started off pretty dumb. So this is where we are, so this is why we moved to Barcelona, because there is nowhere else in the world outside of California where you can do research like this. And it has been a great place to work. It's really, I think it is now, Barcelona, there are a few institutes there which are as good as anywhere in Europe, and I hope that it will continue and that more and more people will move there. Okay, so this question is like, where do the characteristics of humans, with animals, this amazing diversity of life, where does it come from? And obviously genetics is part of this. So if you take two humans, these are two phenotypically quite similar humans, kind of white Anglo-Saxon aggressive males, and they have 8,000. The 8,000 of their proteins are different. Intelligent males as well, and so we've been kind of playing with this idea of, so if you have a human genome sequence, can you make predictions from that genome? And we've been trying to do this not using humans, but using the organisms that we understand best, and this is the unicellular yeasts. So if we sequence yeast individuals from around the world, can we actually predict how they're different from their genome sequences? Okay, the other reason, of course, why individuals are different is because we do bad stuff to ourselves. But the problem we've really been focusing on that I wanted to study when we moved to Barcelona is this. So now this is C. elegans, so this is our favorite organism. So these are 3 developing C. elegans embryos in the real time. So they're genetically the same apart from the per generation mutation rate. They have a deletion of 1 gene. They had the same parent. So this animal, this hatches and has the wild type phenotype, so it's going to be a normal worm. This animal hatches with a mutant phenotype, so it's a sort of ugly worm. And then this animal is going to die straight after hatching. So if you give this talk in England, everybody at that point goes, oh, and feels very sympathetic to the worm. So it's a good test of a nation. This is this question, which is a very old observation in genetics, that when you have individuals which are carrying the same mutation, not all of them are affected by the mutation, or they're affected to different extents. So this mutation which I'm showing you is a deletion of a gene in C. elegans. So this is, you're controlling the genetics, you're controlling the environment, you delete 1 gene which you inherit as deleted from your parents, and the outcome seems variable still. So for this mutation which we study, which is the reason we study it, is because the outcome is 50-50. For other mutations, the outcome may be 90% or 10%, etc. So 50% of them are fine and will continue the line, and 50% of them will die. And the question is, what goes on? Why does the same mutation have different outcomes in different individuals? So, okay, so C. elegans is a remarkable beast of an animal because at the cellular level, its development is the same from animal to animal. So this is a developing worm where you can see the nuclei just colored by depth. And this happens the same over and over again. Okay, so now it starts to form into a worm. And each of these cells, we know what it's called, we know when it will divide, we know what it will become, we know whether it will move. And it's the same from animal to animal. And you can actually do this. So this is a computer version of this. This is real data. So these are the cells just colored by their ultimate fate. So the ones in red, for example, are the intestine moving in. And this is the same from animal to animal. So it's this extreme example at the cellular level of invariance in biology. It looks the same, one from the other. But then when you make mutations, when they have these detrimental mutations, then you have this variation which appears. So where does this variation come from? So if you just look at the animals, they all look the same. But the point is, if you go to the molecular level, so you look at the expression of genes or proteins, then they're not the same. Okay, so this is a gene which is quantified, that's expressed in the mouth of the worm. And you can see that some of the animals have higher expression than others. Okay, and these are all genetically identical animals in the same environment. But the gene expression, you cannot control it precisely. Okay, and biology is not this precise, extremely controlled thing. There is always some sloppiness in the system. So you can quantify this, and not all the animals are the same. So the kind of idea is, and this idea comes from work in yeast and bacteria, is that perhaps this variation in the amount to which genes are turned on and off in individuals is one of the causes of why a mutation will kill one individual but not another individual. Okay, so it could be because of this variation in gene expression. But the point is, C. elegans, like humans, has 20,000 genes. So if we're interested in one mutation, what gene should we look at? Okay, what do we have to measure about the system to predict what's happening? And this is when we had this idea of going back to genetics, because we know how to study which are the genes which are important for any mutation. So this is a very old idea in genetics. These are enhancer screens, epistasis, genetic interactions. So you can take a worm which is deleted for one gene, and it may be okay. And then you can inhibit the expression of other genes one by one and say, does the inhibition of this second gene make the effect of the mutation stronger or weaker? Okay? And these are very abundant things. These are epistatic genetic interactions, and especially in yeast, these have been mapped on a massive scale. So our idea for what was going on so that's looking at how does two mutations interact, okay? But our idea of what's going on when you just have a single mutation is that you have one gene which is mutated, and then you have the second gene, which is the genetic interaction partner, the epistatic interaction partner. And rather than being mutated, it's just varying in the amount to which it's turned on or off. So it's a quantitative effect. So in this toy example, so we have the first gene, which is a mutated gene, and then we have the second gene, which is the genetic interaction partner. So we know when we delete this, it makes the effect stronger. And if in one individual this gene is turned on at the critical moment very high, then the animal is okay. But if at the critical moment this gene is just not quite expressed enough, okay, and this could be a stochastic effect or an environmental effect, then you have one gene which is deleted and the second gene which is not made enough of it, the protein or the RNA, and therefore you have this equivalent of this double mutant, okay? So you have one gene deleted and the one not enough, and therefore you get a synthetic lethal, so a genetic interaction. So the whole idea of this is not mechanistic, okay? This is not mechanistic biology, but it's just a way that we can, for any mutation, we can use genetics to ask what are the important other genes, and then we can see, do those genes actually vary from individual to individual and how much they're switched on and off, and then we can use that to go in and predict what's actually going to happen for each individual. So I'll show you some examples of this that we've been testing in C. elegans. But the first thing I want to say is that this question of how do genes interact, so how does one mutation affect another mutation, this has been mapped on a massive scale, but is still very poorly understood at the sort of molecular mechanistic biology way. So people tend to think about mutations as genes being redundant with each other. This is a very simple biochemical model, but most genetic interactions aren't like this. They're happening between different bits of the cell which don't make much mechanistic sense. Okay, but I'll show you the one that does make sense. So this is the example of when you have, so this is the gene that we delete, okay? And this gene has a very related biochemically functional gene which comes from an ancestral duplication. So this is the example of two partially redundant genes, so two proteins which can do similar things. And this is a very common thing in biology. And so the idea is that, so this is this mutation that we're interested in. And what we want to do is we want to test the idea that one of the reasons why this mutation sometimes kills animals and sometimes doesn't is because the expression of this second gene varies. Okay, so it's a very simple experiment. We delete one gene and we measure the amount the other one is making during early development. And then we ask, does this predict what's going on? So this is how we do the experiments, and this is why we do it in C. elegans, because C. elegans embryos, they grow outside, they develop outside. You can have very large numbers of them, and you can image them as they're developing. So these are embryos developing, and then at the same time as they're developing, we're quantifying how much each gene is turned on, or the genes we're interested in. So you can see here, this is the amount of expression of a reported gene, okay, just fluorescence. So in these plots, so this is time, so this is from the 4-cell stage of development to partially through. And then each one of these is the trace from an individual embryo about how much this reporter is turned on or off. And these are all normal embryos under normal conditions. So you have this big variation. So not all of the embryos as they're developing are expressing the same amount of this reporter construct. So now we can use this kind of system as we can go in and we can delete one gene and we can measure how much of another gene is being made. Okay, so now these are all developing embryos and we're quantifying quite precisely gene expression. And then you let time go forward and you say, does this quantification actually predict what happens to each embryo? So does each of these embryos, retrospectively, did it die? So this one has already died, or does it carry on developing? And does it hatch as a normal worm or as an ugly worm? So you can see them all hatching. And this is all done in a very low-tech way, I have to say. So Alejandro, who did all of this, designed the system. It's all done by hand. There's nothing particularly sophisticated apart from the imaging. Okay, so when we do these kinds of experiments, the first thing to say is that what happens in the mutant scenario is not the same as in the wild type. So this is the expression of the gene in green in the wild type animals, the normal animals. And in blue, this is the expression of the same gene in the mutant background. Okay, so the expression compensates, it goes up. So there's a negative feedback. So when you have a mutation, the system responds. And now we can ask the situation of what happens though. Now these are all genetically mutant animals, and they're just color-coded by what happened to them later on. Okay, so the ones in red, these are the ones which are genetically mutant and phenotypically mutant, and the ones in blue are the ones which were genetically mutant but phenotypically normal. Okay, so there's three interesting things here for this example. Is that if this compensation, so if the expression of this second gene is very low, which is quite rare, then you always develop the mutant phenotype. Okay, so you have one gene deleted, another gene you're not making very much of it, and therefore you die. But if the expression of this second gene, which is still rare, is quite high, so there's very strong compensation, then you always develop with the wild type phenotype. So you have one gene deleted, but the second gene gets upregulated very strongly, and therefore the animal survives. And most animals are just in the middle here, where there is a bias to what's going on, but it's just a bias. Okay, so the most important conclusion from all of this stuff, which is we knew before we even started these experiments, is that from model organisms, so from mice, from worms, where you can make them isogenic, so they're all genetically the same, they're identical twins, and you can control the environment, you often can't predict what's going to happen. Okay, so this is what inspired this work. But this is a very important message for genetics and for biology and for human genetics in particular. And then we think that one of the reasons of why this happens is because during early development of animals, we all start from one cell with small numbers of molecules. And then you have these differences at this stage. And these small differences in gene expression, the extent to which things are being turned on or off, when you have detrimental mutations can be important. Okay, so we think basically that one of the things that's going on here between this early embryo and whether you survive or whether you die is the variation in expression of one gene. But there could be many genes which are important for a particular mutation. And these could be very specific things, so very biochemically related genes, or they could be different parts of the cell. It could not be gene expression, it could be signaling, it could be mechanics of the embryo, which vary. So any kind of source of variation that comes in. So I'll show you one other example, okay? So I'll show you the opposite extreme. So I showed you a very specific example, and now I'll show you a very general example of how this can happen. So if we look at how mutations interact, so I have a mutation in one gene and a mutation in a second gene, often mutations interact to cause a detrimental effect if they're functionally related to each other, okay? But actually, a lot of mutations interact, and they don't seem to encode genes which are functionally related to each other at all, okay? So this is a toy example, but if we inhibit this black gene, the expression of it, it may enhance the effects of mutations in many different genes with many different functions, okay? So no specificity, sort of horrible thing you don't want to work on. But this has been observed very systematically, and the examples include very general bits of the cell, okay? So if in C. elegans you mess up the chromatin, the gene expression machinery of C. elegans a bit, lots of mutations start having stronger effects, okay? But the example of this we've been playing with is molecular chaperones. So from the work of many people, we know that many mutations are affected by the outcome of molecular chaperones, the activity. So chaperones are just proteins which enhance the folding or activity of other genes. And this is an example, okay? So if you take a young worm or a cell and you give it a little heat stress, what that cell does is it turns on a stress response, or the animal does, it turns on a stress response and makes more chaperones and a stress response. And this means that in the future, the animal or the cell, the human cell, is more resistant to environmental stress, okay? This is called hormesis, which has quite a bad reputation because it sounds a bit like homeopathy, because you're giving a little bit of something to protect against a lot of it, okay? But it does actually work very reliably. It's also likened to Nietzsche, so this is German philosophy, so that which does not kill us makes us stronger, which if you went to school in England, to a traditional school, that's the kind of philosophy of English education as well. Anyway, so but the point is that we know, this is just an example, but the example is that we know that animals do not always have the same chaperone content in them, okay? So if you stress them, they make more chaperones. And we know that many mutations, the outcome depends on chaperones. So most mutations destabilize proteins or are somehow indirectly dependent on chaperone activity. And so if you have an animal, so these are just 3 different mutations in worms that have an incomplete penetrance, so they don't affect all the individuals. And the only difference between these animals is that the ones in gray, they received a mild heat shock before the point at which the mutation acts. So what happens is you give the animals the heat shock, they upregulate chaperones, and this means they're less likely to be affected by the mutation. Okay, so the penetrance drops down. And this we think is quite a general thing. And it's an example of how the outcome of your genome can depend on the environment in quite a general way. So not these very specific gene-environment interactions, but an example of how changing the environment can actually quite generally affect the outcome of mutations. And what's more about this kind of thing is that the way that animals respond to the environment, it varies as well, of course. Okay, so these very fast environmental responses are very variable. And so if you have animals which have a strong stress response and animals which have a weak stress response, then the ones with the strong response are less likely to be affected by bad mutations. Okay? But all of that is looking after an environmental treatment. So how does the effect of the environment influence mutation outcome? But to get back to the thing we're really interested in, which is what happens is early on in development, is the question is, early on in animal development, is there also this variation in this general cellular machinery? And so we looked at this. So these are developing embryos where you can see the transcription, the expression of HSP90, which is probably the most abundant chaperone in the embryo. And you can see that there's this big variation. So some of the embryos have much more of this transcription of this protein than others, okay? So you can quantify this and you get this variation. So this means that, this is very surprising to us, that during embryonic development, for some reason, some embryos kind of have this stress response already in, and some of them don't, okay? And this is something we're trying to understand. And then we can play exactly the same games. So now we can delete a gene, that is chaperone dependent, the mutation outcome, and we can measure the expression of the chaperones, and then we can see what happened to each embryo. Okay? So here, the ones if you have very low expression of the chaperone, there are more red ones here, which means there are more of the phenotypically abnormal ones, and if there is high expression of the chaperone, there are more blue ones, which means there's more wild-type protected embryos. Okay? And this is just normal variation of this protein in a population maintained under very nice conditions in the lab. Okay? So there is this variation in the chaperone content, and it's predicting to some extent what's happening. So to summarize for this model mutation that we've been working on, we think there's at least two things going on here. So one is variation in the very precise bit of biochemistry related to the function of this gene. And the second thing is this variation in this general state of the cell, the stress response state of the cell. So now we can ask the question, okay, so we have two things, so we can go in and measure both of these things, and we can ask, does it predict better? So we're measuring the expression of the molecular chaperone, so the general thing, and we're measuring the expression of the transcription factor, the specific thing. And if you compare across individuals, these are not related to each other, so you can be an individual which has high expression of both of these, or you can be an individual which just has high expression of one of them. And so then we can ask, well, what happened to each of these embryos? So we've quantified as they're developing, and what happened to these embryos, or what happened to these embryos? And one way to look at this is we just divide the population up into 4 on the median. So we're deleting one gene, and then we're quantifying these 2 other genes early on in development. If you're below median expression for both of these, so this is about a quarter of the population because they're unrelated, then 1/3 of the embryos survive. But if you have high expression of, say, the chaperone, but low expression of the transcription factor, so above or below median, then now about 2/3 of them are surviving, and it's quite symmetric for this system. Okay, so if you're high expression of the transcription factor but low chaperone expression, then 2/3 survive. But the really interesting ones are these examples here. So these are embryos; they are all carrying this deletion. But they have high expression for whatever reason we don't understand of this transcription factor, and they have high expression of this molecular chaperone, okay? And now, this is about 1/4 of the population, and now 90% of these embryos survive without any detrimental phenotype, okay? So we've gone from a 50-50 outcome to probably where within experimental error, technical variation, these are all surviving, okay? So this is really epistasis in the way that Bateson first used the word very early on, because now the expression of these 2 genes is masking the effect of this mutation. So if you're a worm and you inherit this mutation, and by chance or for environmental reasons, the expression of these 2 genes is very high, then you won't be affected by the mutation, okay? And this is before you see the environment. This is nothing to do with your growing up or anything like that. It's just the embryonic development and what your parents put into the eggs. Okay, so to really summarize this, that mutations do not have outcomes which are predictable often, even if you control everything. But if you go into the system and measure the system, then you can make accurate predictions. And this is what we think should be done for human personalized medicine, is that we know for many common human diseases from twin studies, you'll never be able to make very accurate predictions from genome sequence alone, okay? You will always have to sequence the genome of an individual and then measure something about them that is correct and can be interpreted in light of the genetics, okay, to make accurate, useful predictions. So if you return to the important question of why did Messi turn out how he was, so the thought experiment is clone Messi, reproduce his environment exactly the same, so take him from Argentina to Barcelona, give him growth hormone, train him, etc. Would you always get still the same Messi at the end? So even if with the genetics is the same and the environment is the same, would you still get exactly the same characters and phenotypes of the individual? And our thinking is that at least in invertebrates, this is not the case because you have the genome sequence and you have your environment, but you also have your parental contribution to the egg and the sperm and the genetics of your parents, and you have this inter-individual variation during development, and you have a properly stochastic thing, which is somatic mutations, so mutations which happen after non-inherited mutations. So this is going on every time a cell divides, and this is the primary cause of human cancer. And it's this combination of these things which actually gives the characteristics of individuals. So just by considering one of them in isolation, we're never going to be able to make accurate predictions about health or disease for complex traits. Okay, so I hope that wasn't too fast or too much science for this, but so this is where we are. It's a fantastic place and a great institute to be, very international. Nearly all of this work was the work of Alejandro, who was the first PhD student in the lab. So all of the work in embryos, and then all of the work on the stress responses and the hormetic effects. And this variation was the work of Olivia, who was one of the first postdocs, who now has her own lab. And then these are, I think it's a complete list. So this is all the people who've worked in the lab since it started. So about half of them are still there and half of them have moved on and to better things. And so these are the people that really deserve all the credit for this. And this is the funding agency. So one thing about working in Spain is you have lots of little grants, okay? So it's a bit like being a Formula One driver. You have to kind of stick branding all over you in all different points. And of course, thank you very much for letting me talk.